The Wyss Institute works on technologies to make biology easier to engineer, and one of those technologies is DNA sequencing. And so we've been working on ways to make DNA sequencing much cheaper. The limiting cost right now is the ability to synthesize individual building blocks, so small, short pieces of DNA called oligonucleotides that range from 20 to 200 base pairs, to stitch them together into more gene size like fragments, which are more on the size of about 1,000 base pairs. And the issue with doing that is we want to be able to harness a source of DNA which is very cheap. In order to get the cost down, we utilize this technology called DNA Microchips. This was a technology initially used to essentially do genome-wide studies where they print the microarrays on a flat surface and then you hybridize to natural sequences, whether it be DNA or RNA, to understand how gene expression is working. Instead of utilizing it as a diagnostic source, we're actually utilizing the raw materials, the DNA on the chip, to provide a low-cost input to our gene synthesis process. What we've shown in this latest paper is that process works really well and it's very reliable, and we can use it to build thousands of individual genes from a single chip at a much lower cost than we were able to before. We want to use these kind of gene synthesis technologies to test hypotheses that we have about the natural world, and also to build engineered systems that let us to do new things. So as examples of scientific things that we're doing, we're looking at how individual genetic elements interact with one another to make particular phenotypes. So what gene synthesis allows us to do is test many thousands of these hypotheses all at the same time. And we combine them with our other high throughput technologies in gene sequencing to really read out, to essentially do multiplex experiments, or in other words, be able to do many hundreds or thousands of experiments in a single one-pot reaction. The second thing we're really working on is how do we harness nature, or the natural world, where we have thousands of sequences that we know about because of the broad reductions in costs of DNA sequencing, to really understand what are the functions of these individual genes. And one thing we can do is just synthesize them. So say we have a particularly interesting class of enzymes. Say they are cellulases, which break down cellulosic materials into sugars that can be used for fermentations in industry. And we could, for example, just synthesize hundreds or thousands of these genes and see which ones work best from the natural world, rather than having to disparately go around the world and search for them. These are the kinds of things that we're thinking about doing.